Hello, my name's John Moore and I'm the R&D Manager for Timberlands and also the Chair of the Technical Steering Team for the Resilient Forest Program. It's my pleasure to introduce the next session of this conference, which is focused on enhancing productivity and wood quality. This is a topic that's of real interest to me. It's something I've worked on for the last 15 years. Unfortunately, we can't be here in person today for this conference. We're doing it by video and there is no field trip, so I thought we'd at least have a small excursion out into the forest before we start.
As an industry, I'm sure that we all share similar aspirations to increase the productivity of our forest estates, as this is a key driver of profitability, while also ensuring that the wood resource we grow meets the needs of our current and future customers. We also need to ensure that our forests are resilient in the face of future challenges that we are likely to face under climate change.
The research being undertaken by Scion and the Bioprotection Research Centre at Lincoln University is critical for positioning the forest sector for the future. The research is providing new insights into the biology of forest growth, identifying new opportunities to increase the productivity of stands, and informing the strategic direction of individual forest growers.
This session contains a diverse range of topics. Loretta Garrett from Scion will synthesise the results from the Puraki Catchment Experimental Area. This catchment was first planted in trees in 1973 and is now coming to the end of its second rotation. Studies undertaken in this research forest underpin models such as sea change and new balm. For us at Timberlands, the levels of productivity achieved at Puraki, which is located close to Kaingaroa Forest, was a driver for the development of our overarching goal to double productivity. The forest has a similar climate to Kaingaroa Forest, but has stands where the MAI is over 50 cubic metres per hectare per year, which highlights the importance of nutrition in lifting productivity. In addition to the legacy of trials such as those at Puraki, we've also seen the development of major new trial infrastructure, such as the Forest Accelerator trials and mid-rotation trials. These trials are investigating novel ways to raise productivity that are based on identifying and overcoming site-specific limitations on productivity. These trials have also provided an important resource for testing new tools and sensors for capturing information on soil properties, climate, and tree growth. Dr. Simeon Smale from Scion will provide an update on the latest results coming out of them. The application of fertiliser to forests has largely been undertaken to treat chronic nutrient deficiencies. One of the challenges has been demonstrating and quantifying the response of stands to applied nitrogen fertiliser. This knowledge is critical for justifying fertiliser programs, as we generally don't realise the benefits until harvest. Loretta Garrett from Scion will present new research which combines techniques such as machine learning and stable isotope analysis that is providing better insights into stand responses to nitrogen fertiliser application. Her presentation will also provide an update on the nutrient balance model, NewBalm, which predicts the supply and demand of key nutrients required by trees. Such knowledge is needed to inform nutrition programs so that we avoid applying excess nutrients, which has a financial and environmental cost. For many years, forestry has largely focused on the trees. Our knowledge of microorganisms was limited to mycorrhizae, as we understand the important role that they play in nutrient uptake by trees. However, there is now a significant body of work underway looking at how other microorganisms, such as fungi and bacteria, that are associated with trees can enhance productivity and tree health. Dr. Helen Whelan from Lincoln University has been studying an endophyte called Trichoderma for its ability to enhance growth as well as improve disease resistance, and will provide an update on her research. Dr. Steve Wakeland from Scion is studying the role of the tree microbiome in productivity and disease resistance. This is a new area that is receiving a lot more attention internationally. It is complex, but as we learn more about it, we'll identify opportunities to enhance its potential to improve productivity and resilience to current and future pathogens. To this end, it's great news that this research area was successful in receiving funding from the latest MBIE Endeavour round, which will provide much needed resources. Finally, I hope you get a lot out of the presentations in this session. I'd like to acknowledge the presenters for all the effort they have put into pre-record their presentations. There will be an opportunity for a question and answer session at the conclusion of all the talks.
Well, good morning, everyone. Today I'm going to summarise the Trichoderma radiata pine research program that is based at the Bioprotection Research Centre at Lincoln University. Today I will concentrate on the contribution of Trichoderma to improving the growth of trees in New Zealand forestry. The Trichoderma radiata pine research program is looking to use beneficial Trichoderma root endophytes to enhance growth and suppress disease in forestry, nurseries and plantations. And if we are successful, this will lead to reduced chemical use and improved growth in nurseries. It will generate healthier forests with faster growth, and we may be able to get protection against diseases not here yet. The Trichoderma inoculants offer a non-chemical, a natural alternative for growth promotion and disease control. This is becoming increasingly important today and in the future, with the emphasis on minimising environmental impact and finding more sustainable ways to operate. First, I'd like to describe what Trichoderma root endophytes are. These are naturally occurring fungi that are found in the soil and in plant roots, and they have a symbiotic relationship with plants, so both the plant and the fungi are receiving benefits from each other. Now, hundreds of Trichoderma strains were harvested from the roots of very healthy plants, native cultivated plants from all around New Zealand. So not only from radiata pine trees, but also from other crops and ornamental and weed plants. They were tested for growth benefits and disease suppression in many greenhouse and plantation trials over a number of years. And this was the work that Dr. Robert Hill did. I'd like to point out that trichodermas do a similar job to mycorrhizal fungi that you'll be familiar with. So trichoderma have been shown to promote plant growth in numerous research or crop trials. It can improve nutrient uptake. It can increase the plant's tolerance to stress like drought. Trichoderma can also activate the plant's defense system and can be aggressive against pathogens. Now, the main difference between Trichoderma and mycorrhiza is that the mycorrhiza are generally seen on the outside of the roots, as you can see in the bottom slides. However, Trichoderma is generally inside the roots and is not visible to the eye. So if you look at the top middle slide, I have an image of Trichoderma hyphae inside a piece of radiata root, and it has been stained with a fluorescent yellow-green dye. Now, luckily, Trichoderma is easy to propagate and grow in the nursery, and I can now get very high yields of spores with my methods. Trichoderma is easy to use in the nursery, with the most efficient application method being as part of a seed coat, although you can apply it as a soil drench with conventional sprayers to the soil, to soil beds or to trays. Recently, I have been getting increased interest from the forestry nurseries to use these trichodermas, so this has been satisfying. Next, I will look at the impact of trichoderma on tree growth in plantations. In winter of 2018, I set up 8 large validation trials, each being around 2 to 3 hectares in size. And the 2 most effective trichoderma treatments were selected from a series of national trials. and these mixtures were named PR6 and PR3A, and they were compared to an untreated control. The trichoderma was applied as part of a seed coat in the nursery, and these seedlings, 1-year-old seedlings, were planted out in 4 regions: Gisborne, Northland, Nelson, Bay of Plenty, Waikato, and within each region there were 2 sites. The trials had a robust statistical design with large plots of 9 by 9 trees, and I have been sampling the middle 5 by 5 trees. And there was also a good level of replication with 7 to 10 reps per trial. Now, last year at the conference, I presented the data for each trial individually, and I showed that there were large, significant increases in height to trichoderma in all regions, apart from Nelson. So when this data was averaged over all trials, we found a significant 6 and 7% increase in height with trichoderma compared to the control. So this was a very promising start. I have just finished doing the 2-year measurements a few weeks ago, and I found a similar result to last year, with tree height being significantly increased by around 6% with trichoderma compared to the control. I also measured trunk diameter in 4 of the trials this year, and I found a larger significant increase to trichoderma of 9 and 10% compared to the controls. So we can conclude from these results that trichoderma mixtures, these 2 trichoderma mixtures applied as a seed coat in the nursery, are giving a significant boost to early growth in the plantation. I'm also starting to measure foliar disease in these trials, and this graph is a result of one of the trichoderma trials where the PR6 trichoderma mixture significantly reduced Dothostroma by about half. So trichoderma, it's not eliminating Dothostroma in this case, but it's keeping the disease at a much lower level. Now, there's been strong interest from the South Island forestry companies. So another validation trial has been established in Nelson this winter using their own stock, and 5 other validation trials will be established in Canterbury, Otago, and Southland this coming winter, in winter 2021. I have also looked at the impact of trichoderma on plant growth in nurseries. I have run 2 trials at the Tainai nursery in Rotorua using 2 important radiata clones that do not root well. And I found that the production values, the number of plants that could go out the gate, was significantly increased by between 45 and 75% compared to the controls, which were with and without fungicide. And this was mainly due to increased survival and root collar diameters at harvest, and also to improved root initiation and growth during the season. I also ran 2 trials at the Lincoln University nursery and found that survival and growth was significantly increased in Douglas fir seedlings, with up to a 10% higher survival rate at harvest and up to a 25% increase in height and a 42% increase in overall plant dry weight. There was also a trial at the Southern Cypresses nursery where I found that trichoderma mixtures resulted in up to a 27% increase in root dry weight in cypress seedlings. So overall, this suggests that trichoderma application can result in higher production values in the nursery, and these larger plants will lead to better establishment in the field. Now, other work that I'm doing is I'm working out whether trichoderma can control canker in cypress plants, and also looking at whether trichoderma can control Swiss needle cast in Douglas fir. I'm looking at the impact of nursery chemicals on applied trichoderma strains and whether the chemicals reduce the colonisation of trichoderma in the roots, because it may be that some nurseries wish to continue using some chemicals to reduce risk, so we need to know how sensitive trichoderma is. Another important issue is, can trichoderma be applied to established forests to increase growth and improve health? A large portion of the New Zealand forestry estate has had no trichoderma, so is there a practical way of applying trichoderma to these forests? Another project is, can trichoderma be used to boost poor soils in nurseries before being brought into production? Just got a pilot trial has just started with this. And lastly, how well does the applied trichoderma persist in the forest? And this is an ongoing project as well. So in conclusion, the two selected trichoderma root endophyte mixtures have so far been validated with significantly improved plant growth and health in the nursery and plantation trees. Now, these trials will be monitored in the future. Early indications are that trichoderma inoculated plantation trees may have less foliar disease than untreated trees. Again, with more monitoring work will be needed here. The selected trichoderma mixtures appear suitable for the 3 forestry species that they've been tested with, and should be appropriate for other species as well. So this means that one mixture may be sufficient for the forestry industry, so this is a bonus. Nursery seed coating or drenching is the most efficient and effective way to introduce trichoderma into the forestry system. And so overall, these trichoderma inoculants appear to be an effective growth and biocontrol tool for the New Zealand forestry industry. And this has led to a business case being currently developed for one of these trichoderma mixtures with a commercial partner. And lastly, a big thank you to the following companies. I'm highly reliant on help from these companies for access and to help establish and monitor the trials, so their help is greatly appreciated. Thank you.
Forestry, a major export industry regionally vital for economic development. Forests are sustainable, protecting the environment, the air, the water and the land. Forests, a place to play and a thriving investment for generations to come. Love our forests.
Hello, my name is Simeon Smale and I'm from Scion. Today I will be presenting some recent research from the Accelerator Trial Series and the Mid-Rotation Trial Series. The Accelerator Trial Series. This is a series of trials, 6 of them set up around New Zealand, with the intent of understanding what are the limitations to productivity at these sites and how can we get through those until we reach the physiological maximum of what we can do, what we can achieve with Pinus radiata at these sites. Now, we have a number of elements in these trials, including variations in the genetics of the trees, looking at different levels of fertilizer application, but we also are looking at novel biostimulant products. We've integrated nursery management and looking for nursery legacy effects into the trees during their production. We're looking at the effects of site establishment and other physical site management practices. We're looking at the soil microbial community that's present at these sites and how it responds to the treatments. We're also looking at silviculture, for example, variations in stocking regime. We have a climatic gradient across the sites, so that gives us the opportunity to look at things around drought tolerance. And we're also bringing to bear the latest modeling and the latest remote sensing technologies as well, so that we can really understand what's going on these sites, how are they responding, and how can we get more out of them. So now we're just going to talk about some results from the Southern Kangaroo Accelerator Trial, which is the oldest trial. It was established in 2015. Now, we've characterized the site as being lacking in nitrogen, particularly in terms of an organic matter pool. What our target is for this site is to increase the size of the organic matter nutrient pool to improve the current crop, but also to improve the site for future rotations as well. And what we've done with New Barn is that we've identified that if we want to reach our productivity target, we need to get an extra 500 kilograms of nitrogen per hectare into this forest. To date, we've added 20 kilograms of nitrogen per hectare as Biuret, and we've compared that to 100 kilograms of nitrogen per hectare added as urea. The Biuret has been added in 3 applications to date, to total that 20 kilograms of nitrogen, whereas the urea has been added in 2 additions of 50 kilograms per hectare. Now, in terms of the growth that we've seen from this, we have significant gains in DBH. Again, you can clearly see that the urea treatment has outperformed the Biuret treatment, but since we've added 5 times as much nitrogen, that's to be expected. But the relative gain, we see the difference isn't 5 times as much. So we think Biuret is performing reasonably well here. In terms of foliar nutrition, again, almost the same response, where urea has produced the greatest increase in foliar nitrogen concentrations. But when we look at phosphorus, we're really seeing a dilution effect come through, that basically the foliar nitrogen is basically diluting out the foliar phosphorus as the tree needle mass increases. Interestingly enough, we're not seeing that same dilution effect to the same extent with Biuret, but we will keep monitoring this one through time. Now, looking at Rangipo. Rangipo is a different kind of site. It's actually a relatively fertile ex-farm site. So the opportunity that we looked at here was, well, how can we get the best possible out of this relatively fertile site? So we looked at the site preparation techniques that were going to be applied and also stocking rates. Now, the conventional treatment for this site would have been to rip it and to stock it at 833 stems per hectare. So we have a no ripping treatment as a comparison, and we also have treatment plots that are stocked at 1282 stems per hectare. So about 450 stems per hectare difference there. Now, in terms of our initial results, given that this trial was established in 2016, we're seeing a very strong negative effect from the ripping treatment. So that conventional practice is being outperformed by not ripping, just by planting into the pasture as it was. And curiously, we're also seeing on an individual tree basis, the trees have performed better under the high stocking regime than the low stocking regime. In terms of foliar nitrogen, we're also seeing significant differences that are pretty much the same, that the ripping treatment is being outperformed by the no-rip treatment, and high stocking is better than low stocking, and this continues for foliar phosphorus as well. Now, in terms of the explanation for these outcomes, we think that the ripping treatment created a lot of opportunity for weeds to come through. So there was a lot of early competition, but it also seems to potentially have disrupted soil processes. So we're in the process of looking at some of those responses. So While we understand that ripping is absolutely vital in some places simply for frost protection to get the tree crop when they're young up off the ground a little bit, where you can avoid ripping in afforestation practices, this is a pretty clear result that you should perhaps try to avoid it, simply because of the productivity gain that you could get in that early phase. With regard to the differences between the high stock and the low stock trials, we don't have a clear explanation for that, but one possibility is that the high stocking plots, they're totally dominated by radiata pine, whereas in the low stocking plots, there's still some pasture, some weeds, some other plants coming through. So our working theory here is that under the high stocking plant, the soil microbe environment and the soil microbes themselves have shifted over entirely to supporting Pinus radiata. That's really all that they're doing, that's what they're looking after. Whereas in the low stocking regime, you've still got a transition. So the system potentially isn't as efficient because it's looking to support a wider range of plants. So that's our theory, and we're going to do some testing to figure it out. But as it stands right now, the higher stocking treatment is certainly delivering bigger and better individual plants. Now, the other element that we can look at is comparing the stock types. And when I say stock types, I'm talking about the different genetics that we've planted out. And I'm calling them stock types rather than clones, simply because we have 10 clones, we have the Attenuata hybrid, but we also have GF19 seedlings that we've planted out at each trial as well. So now we're just going to focus on the first 3 sites, the oldest 3. And they have 11 out of 12 stock types in common, so it's almost the same thing. But what stood out immediately is that the same clone is the best across all 3 sites, clone 31. And at the other end of the scale, the Attenuata hybrid and clone 37 are also consistently the worst genetics. So this suggests that, well, this was initially a bit of a surprise to us. We thought there'd be a little bit more variation given the difference between Southern Koongarra and Rangipo. And indeed, there is some variation. For example, if you look at GF19, you can see that its relative position has changed from site to site, but we still have this one dominant good performer and these 2 consistent poor performers. Realistically, these sites are very close together geographically, so the climatic conditions, if that's particularly powerful, looks to be potentially the driving force of success here. Looking at the next two youngest sites, both established in 2017, these have 8 out of 12 stock types in common with the oldest 3 sites. And these 2 sites are Tairua and Ashley. And immediately we saw some substantial differences to the oldest 3 sites. And for example, the Attenuata hybrid is the second best tree at both sites, whereas at Tairua, clone 37, which was one of the 2 worst performers along with the Attenuata clone, these are now the 2 best performers. However, at Ashley, it is worth knowing that clone 37 is back down in the lower ranks. This suggests that there's a real opportunities for matching the genetics to your site. The limitations can be better overcome by some genetics than others, and there's a real opportunity for interaction here. GF19, again, showed the potential for variation. It was in the top third at Tairua and the bottom third at Ashley. Understanding where and how GF19 responds, we can see it does have a high degree of site specificity. Some consistent, strong performers, clone 31, and unfortunately clone 31 was not available when we established these sites. It would have been great to have it to see where it ranked relative to the Attenuata at Tairua and Ashley. However, what we do have is a situation that we can go and we can explore the drivers. Why is the Attenuata doing well here? Why is clone 37 doing well at Tairua? If we can understand those drivers, we have the opportunity to exploit them as well. So we can utilize them to understand how we can get the clones that we want doing well in the situations that we should be planting them in. But unfortunately, not everything has gone quite to plan. The Tokoiti site, established in 2018, was set up with 3 levels of cultivation. So we basically mulched the original organic matter that was there from the previous harvest and integrated into the soil. And the intent was to increase the moisture holding capacity of this soil. We've dug soil pits post-treatment, and we have seen that we've pulled that organic matter layer deeper. So we've gone from about 30 centimetres to around about 45 centimetres, where we've got a good organic horizon. However, the treatment has also had some negative initial impacts on the radiata crop that we've planted across this site. So that's the levels of mulching. It is noticeable that the growth decrease. So what we want to do is develop a plan to get the stock over this hurdle, because the intention of this treatment is to provide the trees with more water when they are considerably older. So we do want to offset any initial negative effects. So overall, there's considerable scope for added value beyond what we're actually what the intended function of these trials were. The scale and the depth of the data makes them useful for other research, such as the development of remote sensing technologies to remotely characterize foliar properties, simply because the wealth of data we have means we can ground truth those remote observations. And we're also integrating the Ashley site into the Forest Flows MBIE program, and that dovetails perfectly with the drought tolerance work. And now I'd like to move on to the mid-rotation trial series. We have a network of 9 field sites established in 2016 to 17, and they've had treatments in place that include site-specific nutrient balance mixtures, conventional urea, and also foliar application at 7 of the sites as well, using fractions of the dose rates applied to soil. If you consider the nitrogen requirements for a crop, what we've done is using NuBalm and other simulation tools. We've seen if your red line is the current growth, well, what's your nitrogen demand going to be if you push your productivity gain to 20% or 40%? What's the gap that you've got to close? And it shows that you've got to get more nitrogen into the system early on in order to put yourself on that high productivity trajectory. And that's marked up by looking at the annual change in nitrogen demand. So you can see from age about 4 to 10, that's when that peak window sits. And that's really good information to design a nitrogen strategy. But what else might you need beyond nitrogen? So using a study of a range of trial sites where we think we have good and optimal growth, we've looked at the relative ratio between nitrogen and other key elements such as phosphorus and magnesium. And we've done work to understand that if you're going to add this much nitrogen to the system, you're going to need to add this much phosphorus and this much magnesium. Otherwise, you'll simply remove a nitrogen limitation and run into the next limitation, and you still won't be maximizing growth. So this nitrogen quantity that you're adding determines the mass that you need of the other elements as well. And you can see how that's worked in the next slide, where we compare control versus urea treatments versus a site-specific mixture, which is nitrogen and other elements. And that's been our best performer, and overall addressing this deficiencies this way provided a 0.75% volume gain per year relative to the controls, which did outperform the urea mixture. Considering the foliar spray treatment, we have had positive responses to phosphorus applications through foliar spray, and it's very low rates, 0.45 kilograms per hectare. But we do see negative initial responses to foliar nitrogen and N plus P application. Admittedly, it is looking better for N plus P over time. And that's indicated by the fact that those trajectories have dropped below the control line. Now, we've looked at what could be driving this negative response, and we think we can understand it through climatic pressure. The nitrogen addition increased the size of the needle mass quite significantly. And unfortunately, we had 2 periods of drought, and the trees that had that bigger foliar mass suffered more. So therefore their growth rate has proportionally diminished more. However, this is still good understanding, and it does allow us to maybe target this foliar application technology to specific areas. And we're also looking at the potential to include microelements in another foliar spray that's quite commonplace, which is the Dothastroma copper formulation. Just as much as you could, if you're adding copper, there is the opportunity to add other microelements as well. Overall, there are a lot of opportunities in this space. We've got increasing stock rates and seed lots, such as we talked about in the Accelerator Trial Series. It will be placing greater demands on site resources. So we do need to develop these opportunities with industry so that we can get the most out of a site as it grows, as it wants to take more from the site. And the work we've shown that the best time to do that is that age 4 to 10 window. The operational testing of the balanced fertilizer mix does look promising for lifting growth across sites by factoring in the specific demands of those sites. And overall, that 0.75% gain in volume through to harvest could be quite significant. Now, the oldest trials in this series are now 19 years old. That's still a lot of potential growth for them to be augmented through this fertilizer addition. So we will continue to track these to see the extent of the gain. Well, thank you very much for your attention.
Tēnā koutou. My name is Loretta Garrett from Scion, and today I'm presenting on the Puraki Experimental Forest. The outline of the talk will step through some history of Puraki, the legacy Puraki has made, some key findings, and what are the next steps for Puraki. Purukohukohu Experimental Basin is located in the central North Island, just south of Rotorua, and is made up of 3 different land uses, of which Puraki is one. Purukohukohu Experimental Basin was established in 1968 under the umbrella of the International Hydrological Decade, a partnership of 3 government departments: Lands and Survey, the former site owner, the Ministry of Works, Water and Soil Division, and the Forest Research Institute of New Zealand, now Scion, designed and implemented an ambitious program of long-term research to understand the effects of land use change on the hydrological regime. In addition to hydrological research, the Forest Research Institute instigated research aligned with the International Biological Program, focused on productivity and dry matter production in planted forests. The installation of the paired catchment site was visionary in ambition and scope, interagencies, and has longitudinal data sets with value that have compounded over time. By the close of 1968, 59 catchments were installed across New Zealand as part of this representative program. Today, only Purukohukohu is still actively monitored, making the site unique and a very special resource. Puraki Experimental Forest is part of the Purukohukohu Experimental Basin. Puraki and the surrounding catchments were converted from native forest to pasture in the 1920s. Over time, the land was neglected and colonised by Manuka scrub and second growth hardwood. In 1957, the land was cleared once again and restored to pasture for sheep and beef farming. Then in 1973, Puraki was planted in Pinus radiata for the first time. It has remained under pine forest to this day. The land is currently owned by Landcorp, Pāmu, and Scion has a lease agreement to continue operating at Puraki as a research forest into the next rotation. Puraki Experimental Forest was established in response to a call by international organisations to establish long-term trials to monitor the impacts of change. Over half a century later, we need to monitor change and adapt to its challenges have never been greater. Over the decades, Puraki has supported numerous research areas well beyond the original objectives of the site. The legacy of Puraki includes greater understanding of the impact of land use on water yield and quality and soil carbon and nutrients. Within Puraki, we have improved knowledge of forest health, nutrition and disease, growth by genetics, by environment, by nutrition, growth by solar radiation, and the use of LiDAR for remote monitoring. Data from Puraki has been invaluable in the development and parameterisation of New Zealand's forest carbon, nutritional and forest productive models. For example, Scene Change, the Forest Carbon Predictor, and the Nutrient Balance Model, NUBAM. All of these models are highly significant tools for planted forest management and reporting. It has provided new knowledge on needle disease, nutrient limitations to growth, pine genetics by environment interactions, and data for hydrological modelling and water quality. All of this has been made possible by the infrastructure of the Puraki forest and the drive to keep the experimental forest active. One key infrastructure is the Met Tower, which was active during the first rotation with a CR7 logger system. This climate data has supported all research undertaken at the catchment. The Puraki water weir, located at the base of the catchment, has been a key infrastructure for Puraki, and in the first rotation, the paired land use comparisons allowed for questions around water quality and quantity to be addressed. This is one of the major outcomes from this paired experimental basin. The work showed how over a growing rotation, the annual water yield in the stream decreases compared to pasture land use. It also demonstrated that planted forests have the potential to supply water to downstream users during dry spring and summer months when compared to pasture land use. Plus, it is able to regulate storm flow in wet winter months compared to pasture, resulting in reduced peak flows and small floods as much as by 50%. Within Puraki, upper mid-crown yellowing, a magnesium-potassium nutrient deficiency was observed and research was targeted towards tree nutrition. The findings were planting stock showed a large and heritable variation in upper mid-crown yellowing occurrence, and within site soil variation contributed towards the upper mid-crown yellowing occurrence. Detailed measurements were made to inform wider catchment hydrology, such as stem water flow, and to understand the dynamics of root decay and nutrient cycling. Over time, multiple tree biomasses have occurred, of which the data has underpinned the forest carbon predictor and the nutrient balance model. And deep soil sampling has informed on how much soil carbon the site has and supported the impact of harvesting method on soil carbon stocks, with results showing Puraki has a high amount of soil carbon, about 145 megagrams per hectare of carbon in the top 1 metre of soil depth. The experimental trial infrastructure currently at Puraki is made up of 3 key trials: phenotypic selection by upper mid-crown yellow tolerance, genetics by environment for upper mid-crown yellowing, and seed lot comparison in relation to solar radiation. These experimental plots were established using a range of genotypes from unimproved GF2 stock from Riverhead Forest, 400 genotypes made up of Puraki and Nelson clones of contrasting nutritional characteristics, a Puraki control seed lot made from GF7 stock from the first rotation at Puraki, selected specifically for tree health, and 2 commercially available genotypes, GF30 and a high density stock. Another legacy of Puraki is the installation of a national trial series at 14 sites, which is made up of clonal material planted at the second rotation Puraki forest. I will now step through some key findings from Puraki, specifically focused on improving forest productivity and managing nutrients for productivity and water quality. This figure shows the productivity of Puraki's first rotation. The stock was GF7 planted at 2,200 stems per hectare, with a number of different thinning rates from unthinned to 550 down to 60 stems per hectare at end of rotation. We now overlay the second rotation productivity for selected planting stock and compare it to the first. Improved clonal stock of GF30 in the high density breed and the Puraki control GF7 seed lot all show improved productivity. We can see that for all improved genotypes such as the GF30, the productivity has been greatly increased. This table gives you some numbers. on the first and second rotation productivity between planting stock at age 20 years. With improved genotypes and higher stocking rates, Puraki had a 54% increase in productivity in the second rotation when compared to the first. This increase has come about through selecting superior genotypes and planting at high stocking on a high fertile site affected by nutritional problems and disease problems. The next two slides demonstrate that we can have high productivity with low impacts on water quality. This slide shows a nutrient balance model output of tree biomass and forest floor stocks over 2 rotations. The first rotation accumulated 1,011 kilograms of nitrogen per hectare at the end of rotation in the live biomass pools and the forest floor. This nitrogen was sourced entirely from the soil. In the second rotation, half the site was planted at 500 stems per hectare with no thinning. This management held a similar amount of nitrogen on site in that biomass pool. With the improved Puraki clone stock selected for tree health, the nitrogen accumulation increases by an additional 340 kilograms of nitrogen per hectare. And with planting a GF30 stock, a further 534 kilograms of nitrogen per hectare is accumulated in that second rotation. And this is what we saw reflected in that growth data as well. This demonstrates that with improved genotypes and higher growth, the demand for nutrients from the site will also be higher. This slide shows the nitrogen loss in kilograms per hectare per year over the first rotation and part way into the second. If we take the average nitrate loss across the first rotation, the value is less than 2 kilograms of nitrogen per hectare per year being lost to the stream. This research offers the potential to develop persistent, high productivity, high nitrogen forest management systems that have negligible impacts on water quality. The water quality measurements have been reactivated and are starting to now monitor going into the end of the second rotation. The next steps for Puraki are to collect valuable end of rotation data on productivity, wood quality and water quality. And with coming to harvest age for Puraki, we are now thinking about what the third rotation will look like. We are keen to maintain the visionary approach from 1968 with multi-agency collaboration, long-term forest research trials of international importance, and extend and challenge the boundaries of existing planted forest trials. The results will be the potential for transformative outcomes for forest management. Ngā mihi. Tēnā koutou. My name is Loretta Garrett from Scion, and today I'll be presenting on predicting a productivity response with nitrogen fertiliser addition and an update on the nutrient balance model. So I will present today on two topics: predicting a productivity response with nitrogen fertiliser application and an update on the nutrient balance model. New Zealand planted forests can be deficient in nutrients, and the addition of fertiliser to forests has in the past been to correct nutrient deficiencies and not as a tool to boost productivity. There is now a shift in focus to increasing productivity and building asset value, and fertiliser addition is one tool for doing this. Confidence in knowing if a stand will respond to nitrogen fertiliser is low in the forestry sector. This is due to a large range in productivity responses as a result of fertiliser addition. This figure here shows an analysis of historical fertiliser trials and what productivity responses were measured post-fertiliser application. The results are highly variable, with both positive and negative growth responses. However, if a positive response is going to occur, it will most likely occur within 1 to 3 years after nitrogen fertiliser addition. This uncertainty must be addressed to increase forestry sector confidence when applying nitrogen fertiliser to improve productivity. How do we approach addressing the question of will a site respond to nitrogen fertiliser addition? Data sets from past research are available to investigate why a forest may or may not respond to nitrogen fertiliser. And we have a collaboration with Associate Professor Brian Strahm, who has used nitrogen enrichment factor to predict a productivity response in planted forests in the United States. We then piloted predicting a productivity response using the nitrogen enrichment factor and random forest modelling. In nature, we have heavy and light nitrogen. The ratio of light to heavy nitrogen is used to understand how nitrogen moves from the soil to the plant. An enrichment factor can be calculated using foliage and soil 15N. The more negative this number is, the harder the nitrogen is for plants to access. Past fertiliser trials were sourced, targeting nitrogen fertiliser application of 200 kilograms per hectare, and they also had an archived soil and foliage sample that could be tested for 15N. We developed a random forest model using a number of inputs, including the nitrogen enrichment factor. The results from the model showed tree age and enrichment factor were the two most important variables associated with tree growth, which is periodic annual increment of basal area. And to gain the best growth response with nitrogen fertiliser, target forest stands between 3 to 15 years of age when there is the freedom to grow, and target sites with a more negative enrichment factor where the nitrogen is currently hard for plants to access. To show you how the model can be used in decision making, we have 3 sites at mid-rotation which we would like to decide if the application of nitrogen fertiliser will result in a growth response. The sites range from more negative enrichment factor to less negative enrichment factor, and also include the other site variables to drive the model. This type of output can support decisions on where to apply fertiliser and can also be used in the nutrient balance model to understand on-site nutrient stocks over multiple rotations. To conclude, the random forest model can predict a productivity response with nitrogen fertiliser addition and showed the nitrogen enrichment factor to be of a high importance. Applications and outlook of this work are confidence in productivity gains through site-targeted nitrogen fertiliser application. It could support the next development stage for nutrient balance model to include a site-specific productivity change with nitrogen fertiliser addition. And new data from nitrogen trials will allow model testing and validation. And further model training with new data will allow improved site response predictions and identify limiting factors as to why a site may not respond. Moving on to the second part of the talk for the nutrient balance model update, with a focus on making NuBalm site-specific for nutrient status. NuBalm predicts nitrogen phosphorus pools in the live biomass and forest floor over multiple rotations and can be used as a planning tool to support nutrient management decisions. NuBalm is part of the Forest Carbon Predictor model and has been developed for nitrogen and phosphorus using Puraki forest biomass data, which is a high fertility ex-pasture site. NuBalm has a choice of 3 fertility settings: high, medium and low. The high setting is based on the fertile Puraki forest and the low setting from Kaingaroa forest biomass data, and the medium is simply the average of the two. How it works is, if a medium or low site is selected, an offset is applied to the nitrogen and phosphorus concentrations in the biomass and forest floor components. The offset reduces the biomass component nitrogen and phosphorus concentrations from that of the high setting when predicting forest nitrogen and phosphorus stocks. We wanted to do better than the subjective choice of high, medium and low by using a soil sample from the site to set the fertility input for NuBalm. The following slides describe the FGR funded work to make this improvement. We achieved this by targeting a trial series across a range of soil fertility with clonal tree plantings. The sites were selected based on soil carbon to nitrogen ratio, which is an indicator of low to high fertility. We sampled the soil, stemwood, foliage and forest floor at each of these sites to know how related the soil results were to the stem, foliage and forest floor. If they were highly related, we then had confidence in using the soil to describe the site fertility. Using the soil sample, nitrogen stock estimates from NuBalm performed well for a site with no fertiliser addition. Where fertiliser had been applied, they tended to under-predict nitrogen stocks. It was found that soil phosphorus was unrelated to stemwood phosphorus, however, was related to foliage phosphorus. The result was the phosphorus stock estimates from NuBalm tended to be over-predicted. I'll now show you an example of using NuBalm in decision making. NuBalm has simple inputs of a given 300 index value, excuse me, or a stand measurement, silvicultural management regime, and the site fertility measured using a soil sample. You can use NuBalm to predict your actual productivity nutrient uptake and what the gap in nutrients is required to reach that target productivity. The example here shows the amount of nitrogen in the tree biomass and forest floor over one rotation for the actual productivity and the target productivity. What the model run shows is that there is a 340 kilogram per hectare nitrogen gap. This gap must be overcome from either external inputs or through making better use of what is already there. To conclude, NuBalm is now site-specific for nitrogen. It requires simple inputs of 300 index or stand measurement silvicultural management regime, and a site fertility measured using a soil sample. A very basic version is available on Forecaster. Scion has the latest version and can work with industry to generate predictions. NUBARM is currently being used in treatment planning for the accelerator trials, linking with Overseer, in planning nitrogen mining from nitrogen saturated soils, and there's interest from councils to use NUBARM to support afforestation efforts. Ngā mihi.
Hello, I'm John Lee. I'm a wood quality scientist from Scion in Rotorua, and today I'll be presenting on the DiscBot, which is a wood quality scanning system developed by us. So the DiscBot, a wood quality scanning system. What is wood quality? During wood formation, numerous factors both inside and outside the tree lead to variation in the type, number, size, shape, physical structure, and chemical composition of the wood elements. Wood formation is the process, but wood quality is the result. So what is the DiscBot system? On the left, basically, it's a bunch of sensors and cameras bolted together with a slot pro frame parked on top of a Parker XY moving table. And what does it measure? It's an array of scanning sensors or stations that measure or infer wood quality traits both directly and indirectly. We have X-ray, which measures wood density, NIR or near infrared, which measures wood chemistry, USV or ultrasonic velocity, which is a measure of stiffness or microfibril angle, and MLT, which is masked light transmission, which is basically a bright light shine through the disc and the light follows the fibers, which gives us a grain angle. Here we have a picture of the DiscBot in action. The discs are loaded into the green cassette that is then placed into the robot system. Here we have the USV being measured. The disc passes between two probes which send an ultrasonic sound between them. It covers the entire surface of the disc. The same process is used for all of the other sensors where the robot travels the sample under the sensors themselves. All the data then is automatically just packaged together and sent off to a cloud database. And then a new sample is added. The scanning takes around anywhere between 8 and 15 minutes, depending on the size of the sample. When we talk about wood quality models, in the past, there generally were average models of wood quality. So we would look at a bunch of cores for an area or a site and produce basically wood quality distributions where we had low density at the pith, and it gets better as you get out and with age. We know that there is no average tree. When a tree goes to the sawmill, it has particular bits of peculiarities in it, compression wood, changes in density, changes in stiffness, changes in grain angle, all these things that we can readily see. We also know about growth rate and tree shape and tree architecture. The bit that the DiscBot wants to put into that is to be able to infer that variation so that we can then model trees that are more likely to reflect the actual tree in the forest. Now, when we talk about the real variation, these are the vertical lines. Each one is a stem, 21 stems here. And there's a disc taken every 1 meter up the stem, all on the same site, all the same age. So you can see there's a huge amount of variation in shape and size and distributions. These are a photo image of some green discs just after they've been surfaced. While everything's green, we can see a few things. We can see the heartwood, we can see the ring boundaries, and we can see in this case some patches of compression wood. As the discs dry out, we tend to lose the ability to see these things. And our DiscBot runs on dry discs. There's other factors we of course can't see, the chemistry, the density, the stiffness patterns across those disc areas. These are scanned images from the disc. From the left to right, we have density, the middle one galactan, and the right-hand side is the USV. So we can see the distributions across those. Interesting, the galactone one, the discs that you saw just before this, you can see the compression wood, the light blue area showing up quite well there. Also in the USV, the same area as a dark blue. So you've got high stiffness, but also high compression wood in the same area. That would make some interesting timber. Additional applications being developed with the CoreBot. So basically we've altered it to measure 12 mil cores, and at the moment scanning for only density and the near infrared. We've tried to automate the ring boundary allocation. We're looking at identifying heartwood development in dry wood. And we've looked at small modified wood samples and going to be looking at other species like cypresses, totara, and eucalypts. Here's an example of the CoreBot bark to pith scan, spark on the left, coming to the right. This is lignan, so you can also already see that there's quite a variation. So you can start to actually quickly and cheaply look at trees and make an assessment whether maybe you need to take a full disc or live with some more average properties. Ring digitization. It's usually quite nice to know when the thinning was happened or a fertilizer was put on and what the results of those things were, or if there's been some sort of climatic change that may have affected growth. We can go back by a year. Currently we've been manually doing this. It's very slow, can take up to 20 minutes to do a large disc, and we're prone to human errors where people see things differently. An automatic approach has been developed, fast batch processing using the disc density data. Here in the yellow we have density scans, and what we've done is taken radial paths from the pith to the bark and taken the density data from those radial scans. We've taken those every 5 degrees around the stem. Each of these vertical lines is a representation of those radial strips, and using mathematics and working the front edge of every one of the clumps where it makes sense, we're able to link those together and give us the ring boundaries. Why are we doing this? We'd like to understand the connection between genetics, environment, the site, and the end product quality. So feeding data into our next generation wood quality models. Improve the confidence of processors in the resource coming on stream. Early quality assessments of stands pre-harvest. Provide recommendations on how to grow better wood quality or better wood in the future, and help tree breeders target improved product quality outcomes, and provide a framework for growers and processors to have a conversation about benefit sharing. So what's good for the processor might be good for the grower. This has been a long project, up to 7 years, and a lot of people have come and gone. These are just some of them, and every one of them has had a very valuable input into this machine. Thank you very much. I hope you enjoyed the presentation and have a nice day.
Our planted forests, a sustainable resource. Trees locking up millions of tonnes of climate change surplus carbon from the atmosphere, then storing it in our buildings. Standing forests, binding the landscape, cleaning waterways and absorbing floodwaters. A sheltering habitat for endangered birds. Forests, a thriving investment for generations to come. As places to ride your bike, go for a run, sweat or relaxation among the trees. Love our forests.
Hi everybody, my name is Steve Wakelin, Research Leader at Scion Research. And today I've got the privilege of talking to you all about the pine microbiome projects that we've got happening here in Scion. So let's just go into the share screen here. Stop the video. So hopefully that's coming through quite well. So the title of my talk is The Role of the Tree Microbiome, and that will become clear as we go through the presentation. So this is certainly on behalf of myself that's presenting, but also the Pine Microbiome Research Team in Scion. So a really good question that we should start off with is, what is a microbiome? So microbiomes refer to the entire community of microorganisms that live in a defined environment. So it's the assemblage of all the species that are there in certain habitats. What's really interesting is that each habitat, whether it's a soil or a plant or a person, has its own unique fingerprint of the microbes that are present. So microbiomes can be incredibly diverse and complex in species and functions. And this picture is Winogradzki's column. It's a very good example of this. Lots of gradients of sulfur, oxygen, and energy means that there's lots of different habitats and spaces for the microbes to live in. So microbiomes can also be very simple. And a good example of this is the microbiome of gold grains. So yes, microbes live on gold, and in this case, there's only 1 or 2 species that are very common in these habitats. It's very simple. So microbiomes aren't just out in the environment in the world, they're also in and on us. And so people will, often talk about the Human Microbiome Project, which was started in the US a few years back. And that was really to try and characterize the assemblage of species of microbes that make up a human being and affect our health. So there's something like 100 trillion different microbes that live on or in each person. What's really cool is that for every 1 human gene, there's something like 150 microbial genes, systematically expanding the catalogue of genes that make up a person. And each individual person has their own microbiome. And that's as unique as a fingerprint in terms of the species that are there. And another important fact is that what we know now, unraveling the human microbiome, tells us that something like 90% of diseases has some sort of link back to the microbiome. So these aren't just diseases associated with your obesity and weight and all the rest of it. It's neurological disorders right through to Parkinson's disease and so on. They've got a microbiome link, which we never knew about until we started this work. So plants too have involved with unique microbiomes. They're in and on the plant surfaces. That includes the soils and the roots, right through to the leaves and the other plant structures. And they're functionally important. They're not just microbes sitting there doing nothing. They have a role to play in the success and life of the plant. And some good examples are how microbiomes extend the phenotypic plasticity or elasticity of a plant and environment. A good example is drought tolerance. Microbes can confer drought tolerance in some plants. The microbiome can change the growth rate of plants, whether that's positive or negative. Of course, we want it to be positive most of the time. The microbiome has a role in causing disease, and also disease suppression, nutrition, right through to regulating the onset of flowering in certain plant species. What's also sort of coming through the literature at the moment is the role of the microbiome in and on the plant and sort of conferring new metabolites and secondary metabolite pathways, which sort of changes what the plant does. And these are really, really important. So these are like flavor compounds in strawberries, or the unique factors in honey. New Zealand's agricultural sector, the ryegrass, is dependent on a microbiome to protect it from the grazing of invertebrate pests. So they're really, really important. I suppose it's not surprising, therefore, that there's really a lot of international investment in understanding and harnessing the microbiome of plants. So here's one example of, Monsanto and Novozymes getting together to form the BioAg Alliance, which is something like $100 million investment in understanding or hacking the microbiome of agricultural plants. This means that they're really trying to reduce the amount of nutrients that plants need to grow, and right through to protecting the plants from diseases and pests. So what about trees, and what about Pinus radiata? So trees should really be the priority research for plant microbiomes. I mean, trees have long breeding cycles, so it can take quite a long time to introduce new traits just by the standard selection and breeding approaches that we have. Because they're in the environment for a long time, once you plant them, they're sort of staying there. Trees also require a large degree of environmental elasticity to future conditions. The conditions that we plant them in today are probably going to be quite different in 20 to 30 years' time. And this is not just the biophysical conditions of the environment, but things like diseases and pests, and maybe the way that we manage forest as well. So what we're proposing is that, like with the human microbiome and other environments, the microbiome of a tree is really a new genetic library that we can add to the tree to confer new traits. And whether this is around productivity gains, stress tolerance, environmental resilient forests, wood formation and quality, there's lots and lots of opportunities there, and we haven't even really started exploring these. So the tree microbiome research, what we aim to do is look at the plethora of the microbes that are on and in a tree and the environment that they're in, and particularly the interactions and the outcomes of those interactions of the microbiome, the tree and its genome, and the environment, all of them together, and how they sort of function to alter the plant physiology and the growth, and even the behaviour, the coordinated behaviour amongst the trees and the forests, and how they sort of change the whole ecosystem state of all the geochemistry, so the nutrient cycling and the carbon storage. Now this is quite a challenge, and we've done a lot of microbiology research, but microbiome research is a whole nother level. And, if English philosopher Francis Bacon said that if we're going to achieve results never before been accomplished, then we must expect to employ methods never before being attempted. Fortunately for us, we've got the Human Microbiome Project and some other endeavours to fall back on, but certainly it's very ambitious to go in and start exploring the tree microbiome. But we do need to start somewhere, because the opportunity size is very big. And towards this, we've got two highly aligned programs of work that we'll just quickly run through. So the first is the Resilient Forest Partnership Program, and the second is some announcement of new government funding. So it's through the MB Endeavour program, a grant called the Pine Microbiome. So most folk will be aware that we've just completed year one of the Resilient Forest Partnership Program, and the role for us in this program in the first year was building the fundamental framework for pine needle microbiome research. So we really needed to get our stuff together to start exploring the microbiome and what it does to the plants and the environment. And we did start with needles because of that direct link between needles and disease and the microbes. And if you're losing your needles and they're not functioning very well, you're losing your solar panels for the plant to grow. So in the first year of this research, we've been exploring, field sterile techniques to collect samples from the tree and the forest, returning those back to the laboratory, trying to understand the variability on the tree and the canopy of what the microbiomes are on a needle. So we're in the canopy to select how old the needles are, what really makes a difference when we go out sampling. So, a fair amount of fundamental framework work had to be done. And then in the laboratory, we need to understand, how to extract the microbiome DNA from the needles. So not just the plant DNA, but all the other DNA that's there, and really scale up the way that we do our sequencing. So traditionally, we might have a sample and we sequence them one by one. What we have been able to do this year is massively parallelize this sequencing. So we're typically sequencing 88 samples in one go at the moment. And then right through to the unique bioinformatic and statistical approaches that we need to start dealing with this data. So really, just getting all our ducks lined up. So as we go further into the program, all the basics are there and we can proceed with confidence. So although year one was really about getting the framework established, some really interesting results are already sort of emerging from this work. An example here is the expected amount of fungi, not even the bacteria, just the fungi, that we can expect on the pine needles of a single plant. And there's more than 1,600 species that we've discovered so far. And that's probably the limit for this plant in this environment. Based on the average genome size of the fungi, there's actually 2.5 times more genetic information and genetic potential and novel information just in that genome compared with that of the tree. So if we remember back to the human microbiome slide where every human gene has 150 microbial genes, well, just on this one tissue component alone, we're starting to see a very similar pattern emerge. What we've also found, which was very nice, was that the fungi that are there aren't just in a single small clade of similar species. There's a massive diversity of species. It's polyphyletic distribution of fungi all across the kingdom there. And that means that the range and the potential of traits and DNA and things that these fungi can do it's absolutely immense. We've got single cellular fungi from yeasts, which are known biological control agents for the phyllosphere, for the needles, right through to the typical branching pathogens and saprophytes that we might expect to see in a forest ecosystem. And just sort of scanning through this tree of the fungi that are present on the pine needles, the sharp-eyed people in the audience will notice that there's a group of the Dothidium mosaics. And this is where Dothostroma, the fungal pathogen, sits within amongst this group. So the needles that we sampled were ostensibly very healthy. That was one of our requirements for sampling. And yet the pathogen, or a close neighbour, is there. So it's sort of silently sitting there and perhaps waiting for the microbiome to change or the conditions to change to express a disease outcome. So already, we're starting to come up with some new understandings of where some pathogens might be sitting latently in the forest, ready for diseases to emerge. Year two of the Resilient Forest Partnership Program is really about building on the gains of year one. So we've built those foundations, we know what to do and how to do it now. And year two is really extending what we do on the needles and our understanding into the different plant tissues. So looking within inside the tree itself, the microbiome in the tree, in the wood, the reproductive tissues, the pollen, the catkins, right through to the bark and cambium. So really the microbiome that's in contact with all these other cells and signaling, that's in a really profound position to change the wood formation, wood quality, growth aspects, hormoning, signaling with the plant and the environment. And this is downstream in 5 or so years' time, really what we want to end up with, where we've built up an understanding of the tree and we understand its genetic potential, we understand what the microbiome is on that tree, the assemblage across the roots and the soil and the trunk and the needles all acting together, and how these are expressed collectively against the environmental conditions, so temperature, silvicultural management, herbicide and pest disease, all that sort of thing, how they are expressed against that entire tree and its microbiome to affect things like carbon storage, how to climate-proof forests, how to control pests and diseases or confer tolerance, how to alter wood quality or store carbon in soils. So really for us, it's taking the next step in our understanding of how forests operate and function. So the other program that I'd just like to announce is some new funding that's come through from the New Zealand government into the forestry sector. So it's an announcement of investment of $13.5 million by MB, and this has been supported by the Forest Growers Levy Trust, and we're very thankful for that. So it's a 5-year research program. It's been led by the team here at Scion, but it's certainly multi-institute and international in flavour. And what we're going to do is use Pinus radiata to determine how evolutionary history, how pine co-evolved with microbes to be successful in the past, how genetics, how the ecological context all influence the outcomes of the root microbiome interactions, and extend the tree phenotype. And the target in the project is around climate proofing forestry, but really that's just the one of the many things that could potentially come out of here. So a key outcome for us is the development and establishment of Pinus radiata as a model tree root soil microbiome platform globally. And the reason that we want to do this is that our resources and the amount of time we have available to make these games in New Zealand is quite limited, and we want to be able to harness off what people can do globally. So if we establish pine as the model, like in a crop species that other research labs take up all around the world and publish on, well, they're essentially doing a lot of the work and investment for us, and those benefits, those science outcomes and how we interpret them for New Zealand forestry will flow through, so we can get, it's a case of working smarter, not working harder. Another key outcome will be the demonstration that targeted manipulation of the microbe on a certain tree or a certain cultivar confers reproducible changes in the physiology of that tree. So really demonstrating, without a doubt, that manipulation of the microbiome can change the tree in beneficial ways. This program that the government's investing in with the Levy Trust is explicitly focusing on soils and the soil root microbiome. And this is a really good outcome, because what that means is that it's directly complementing the work that's happening in the Resilient Forest Partnership Program, which is really above ground tissues. So that these two programs really line up really, really nicely. And to us, this is really an investment in the next generation of microbe silvicultural tools. So in some ways, we, like the Human Microbiome Project, we might not know what's going to come out at the end of this, but there'll certainly be some incredible surprises and opportunities. And in the future, we hope to be able to, use the microbiome as another one of the tools that we've got in our silvicultural toolbox, and the plant genetics and genomes and things as well, to manage sustainable forests for New Zealand's future. So that's the end of the talk. I've just noticed that there's a mistake on there. There should only be one @ in that email address, but I'll leave you there with that. And thank you all for your time and attention. And I just sort of recognise that Timberlands have been really great to us, enabling access into their sites and getting their plants to push this research along. So thank you very much.